Transcription. DNA is transcribed to make RNA. There are three different types of RNA: mRNA, tRNA, and rRNA. mRNA is messenger RNA. It carries information from the DNA to the ribosomes to make proteins. tRNA is transfer RNA. It recognizes specific RNA sequences and transports the amino acids to appropriate regions during protein synthesis. It helps out during translation. rRNA is ribosomal RNA. It forms the ribosomes, which are the structures used to make the proteins. Transcription is the synthesis of a complementary strand of RNA from a DNA template. There's nitrogenous base pairing. We always have C going to G and T going to A, but now A goes to U. U stands for uracil. Transcription begins when RNA polymerase binds to the promoter sequence. The promoter sequence is the beginning of the gene on the DNA. Transcription also proceeds in the 5' to 3' direction, just like what we saw in DNA replication. Transcription stops when it reaches the terminator sequence. So transcription stops when the RNA polymerase moves along the DNA until it reaches the terminator sequence, which is the end of the gene. Once we reach the terminator sequence, mRNA is released. So here we see in the picture here that we have DNA, and we're going to use the DNA as a template to make an mRNA, and that mRNA is going to then make a protein. Well, the step that we're doing now is DNA to mRNA. mRNA to protein is translation, and that's what we'll be discussing next. So, here is the DNA strand, and here is RNA polymerase. RNA polymerase is going to bind to the promoter sequence, and the promoter sequence is then going to signify that this is where the gene begins. RNA polymerase moves along, adding nucleotides. So every time it hits an A, it's going to add a U. Every time it hits a C, it's going to add a G. If it hits a G, of course, it's going to add a C. And then if it hits a T, it's going to add an A. But every time it hits an A, it's going to add a U. And it goes along, all the way along, keeps transcribing until it gets to the very end of the gene, which signifies the terminator sequence. And the terminator sequence, again, is the end of the gene. And once we reach the end of the gene, we have now completed transcription, and the mRNA is released. Translation. The mRNA is translated into codons. Codons are a group of 3 nucleotides. Each codon codes for 1 amino acid. There are 64 sense codons on an mRNA that encodes the 20 amino acids. The genetic code is degenerate. Degeneracy of the code means that each amino acid is coded by more than 1 codon. Degeneracy helps out by decreasing the mutation rate. So there are 2 types of codons. Sense codons. Sense codons code for amino acids. Translation of the mRNA begins at a start codon. So the start codon is always AUG. Nonsense codons are when no amino acids are coded. So we have arrived at the stop codon, and translation ends. There are 3 stop codons: UAA, UAG, and my favorite, UGA. Each tRNA molecule carries the complementary anticodon. The anticodon is complementary to the sense codon. So the anticodon pairs with a codon, and the amino acid is transferred to match the codon. So let's put this all together. Here is our codon chart. And again, remember we talked about the fact that a codon are 3 nucleotides. So 1, 2, and 3. So this is a codon. You have 64 sense codons, and each one of them is going to encode one of the 20 amino acids. So here are your 20 amino acids. You have a degeneracy of the code, and remember that means that each amino acid is coded by more than one codon. So here you'll see that we have proline, and proline here is coded by 4 different codons. Okay? Alanine, 4 different codons. Valine, 4 different codons. And of course, our start codon here is AUG, and then our stop codons here, UGA. So, let's go through the process of translation. So, remember earlier we were looking at DNA to mRNA. We said that was transcription. Now we're going to take a look at the mRNA molecule that was released after transcription was completed to create a protein. In the initiation stage of translation, there are components. 2 ribosomal subunits. So remember we have a large and a small ribosomal subunit. We have our tRNA with the anticodon. Remember, we're always going to start with AUG. And the anticodon is complementary to the codon. So the anticodon will have UAC. That is the complementary anticodon. We have our mRNA to be translated, and of course we have some protein factors. Remember, translation of the mRNA begins at the start codon AUG. And AUG is a codon that codes for methionine. So here's the steps. The ribosomal subunits are going to attach. So the small and the large are going to come together and attach to form a complete ribosome at the start codon. The tRNA is going to signal for the correct amino acid, and the tRNA is going to attach to the correct codon, in which we're going to see anticodon to codon pairing. So here is the complete ribosome formed. Here we're going to form 2 sites. A P site, in which the first codon is sitting with the tRNA, and an A site. So a second tRNA is going to come along with an amino acid to attach with the next codon at the A site. So the next codon is UUA. So the anticodon will be AAU. Okay? So now these 2 tRNAs are going to sit in the sites. So this is your P site, this is your A site. Now methionine and leucine are going to form a peptide bond between them. So the ribosome is going to form a peptide bond between the adjacent amino acids. So now they're going to be holding hands. Okay? So now they're holding hands. Now we need to move on to the next codon. So if you see here, we have nowhere to move. So the ribosome is going to move, or what we call translocate. So the ribosome is going to move to the next codon. So here's your next codon, GGU. So here it is. So now we're going to have another site open up, and that's my E site. The E site is the exit site. So now we have 3 sites, EPA. That's my mnemonic, EPA. So exit site is the site in which the tRNA is going to leave. So now GGU, the anticodon of course is CCA, and that codes for glycine. Now notice, methionine and leucine are holding hands with one another because they formed a peptide bond. So what do you think is going to happen now? You are correct. A peptide bond is going to now form between leucine and glycine, and they're going to hold hands. Now, this tRNA at the E site has to leave because now this tRNA has to move into that site, this one has to move into the P site, and this next tRNA is going to come along and code UUU. So tRNA at the E site is going to leave while another tRNA attaches. So we're going to see in this picture, now we're going to move along, and we keep on moving. So the ribosome translocates, so the mRNA is not moving, it's the ribosome that's moving. So the ribosome moves down the mRNA, and new peptide bonds are formed between each one of these new amino acids. And then the next E site tRNA leaves, and the ribosome moves again. And it keeps moving, and a polypeptide chain is going to be formed. And this process continues until the stop codon is reached. So here you see that we have reached our stop codon UGA. So that signifies that we have reached the end of our protein. So there, translation is going to end. All the tRNAs are going to leave. So your new protein that you have made also leaves. And the ribosomal subunits are going to dissociate. So they're no longer going to be complete. The large goes its way, the small goes its way, and everyone leaves. Prokaryote versus eukaryote. In prokaryote, transcription and translation occurs in the cytoplasm. Translation may begin before the transcription of a particular mRNA is complete, since the start codons of the mRNA being transcribed is available to the ribosomes. Prokaryotes and eukaryotes have different ribosomes. Remember, prokaryotes have 70S, eukaryotes have 80S. In eukaryotes, transcription occurs in the nucleus. RNA is processed before translation. In the mRNA, you have exons, which are expressed DNA. You have introns, which are intervening DNA. The introns are removed from the RNA transcript, and the exons are joined by ribozymes before translation, which occurs at the ER in the cytoplasm. So let's take a closer look at this. You'll see that we have our DNA, and our DNA has exons and introns. Remember, your exons are going to be your expressed DNA. Your introns are your intervening DNA. So we take the DNA and we transcribe it into mRNA. And you notice the mRNA has the exons and the introns. What we see next is that we have to process this. And all of this is occurring in the nucleus. So you see the big purple blob? That's the nucleus. Ribozymes are going to remove, the introns so that we form just the expressed DNA together. From there, the expressed DNA comes together, and the mRNA is then going to leave the nucleus through the nuclear pores. And when they leave through the nuclear pores, now they can go to the ribosomes and become translated. Here shows the simultaneous transcription and translation of the ribosomes and the mRNA in prokaryotes. Because remember, everything's in one place. So as I'm making the mRNA, I can also go ahead and translate at the same time. The operon model. The operon has three components. The promoter. The promoter is the DNA segment where the RNA polymerase binds and starts transcription. So here, you see your promoter. The operator. The operator is the DNA segment found between the promoter and structural genes. It determines if transcription will take place. If the operator is turned on, transcription will occur. So here's your operator. The structural gene contains the genetic code. So here's your genetic code. Regulation is important since protein synthesis requires a lot of energy, so it's important to use it wisely. Constitutive versus regulated. Constitutive genes are expressed at a fixed rate. Regulated genes are expressed only as needed. Repression, which is transcriptional level regulation, occurs when we have an overabundance of a metabolite product. So repressible enzymes is usually a response to overabundance of a metabolic product. Repressors block RNA polymerase from initiating transcription of the enzyme that creates the metabolic product. Induction is also in transcriptional level regulation, and that is when there's a need for a certain metabolic product. Inducers induce transcription of the inducible enzyme that creates that product. So if we take a look at this picture, we'll see that normally without the repressor, there's nothing blocking transcription, so the operator is turned on. Okay, and RNA polymerase can bind and transcription can proceed. However, if we have too much of a product, too much protein being produced, then a repressor is going to sit on the operator, turning it off. So when RNA polymerase binds to the promoter, which is the start, it's going to block RNA polymerase from being able to carry out transcription. Hence, we inhibit the production of that product. Here ends Part B of Chapter 5.